The most important thing is I have over 35 years of experience working with both Spiroplasma cunculi and Dabulus matus. Now, the big news for me is I retired in 2021, which was about 3 years too soon, because guess what happened this summer? Guess what happened this summer? I don't know. Oh, gosh, that would help.
There we go.
There you go. This happened this summer. I got a call from Ashley and Mayura, and they said, "Hey, Austri, what do you know about corn stunt and Dabulus matus?" And I just like, what? And Mayura took me out to the field, and I saw the first corn stunting symptoms here in the United States. I've seen it in Ecuador and in Brazil, but not in the US. And I quite frankly was shocked. And that began the beginning of a nice relationship with these two extension agents as we're learning more about corn stunt in the state of Oklahoma. So what am I going to do? I'm going to really quickly talk about the biology of the corn stunt pathogen, Spiroplasma cunculi, and talk about identification and biology of the primary vector, Dabulus matus, and just talk about other leafhoppers in the maize ecosystem, which includes grasslands, and some of the important life history traits, which are important for understanding why this leafhopper is such an excellent vector of this particular pathogen. Talk a little bit about the history and epidemiology of corn stunt disease and evolution of the corn stunt Dabulus pathovar system. Okay, let's get started. These are the two characters. The bacterium, Spiroplasma cunculi, the leafhopper, and of course, when they come together, we get symptoms like this, corn stunt disease in Oklahoma. All right, corn stunt is a disease complex. It's a complex of 4 pathogens, 2 bacteria, and 2 viruses. Corn stunt spiroplasma, maize bushy stunt phytoplasma, maize rhiadophenovirus, and maize stripe virus. These are all phloem restricted. They live in the phloem of maize. Okay, so that's an important characteristic. I'm going to concentrate on corn stunt spiroplasma because this one is by far the most important pathogen in the corn stunt complex. It always shows up. The other pathogens may not be there, but this one always does. Okay, so what do these look like individually? Mayura is going to give you a much better description of these pathogens in the corn stunt complex, but this is what they look like when you separate them out and transmit it to maize. So here is maize rhiadophenovirus. The vector is Dabulus matus. It's this beautiful clearing within the phloem. Maize bushy stunt phytoplasma causes phyllidy and stunting, shortened internodes, and these very bright symptoms in the leaves. Here's an early infection of corn stunt spiroplasma. You can see the clearing of the veins, etc. Later on in infection, you get that really bright red coloration striping in the leaves. These are all transmitted by Dabulus matus. The 4th virus is maize stripe virus. Sorry, the 4th pathogen is maize stripe. And you get similar clearing of the veins here. The difference is the vector is the plant hopper Peregrinus matus. Okay, as far as I know, Peregrinus is not in the state of Oklahoma. It is in the southeastern United States. It's considered cosmopolitan. So even though it may not be here now, you might be able to see it coming in the future. All right, so important to note that corn stunt disease may or may not involve all 4 pathogens in this complex. Corn stunt spiroplasma, as I said before, is by far the most important pathogen because it always seems to appear every time. Would you concur with that? It's always there in corn stunt. All right, so let's get a little bit into the history of this. Corn, and you have to do that by understanding how corn was domesticated. I'm sure you're familiar with this story, but I'm just going to review it really quickly. Corn was domesticated, developed, selected for by the early Mesoamericans in Mexico. Oh, it's estimated between 8,000 and 9,000 years ago. People talk about this, debate it. But so the early progenitors of corn were developed from teosinte in central Mexico. This is what gamma grass as a teosinte looks like now. It's just sort of a bushy grass. It almost looks like panicum. And here are the seed heads. And so those early Mesoamericans, they took this, heated it up. They ate it, but it was rather impalatable. So they they selected for traits that made it more edible. And as they did this, they started to develop more corn-like, more maize-like characteristics. At the same time, there were a group of leafhoppers that colonized and used the early teosintes as a host. These were the dabulous progenitors who were adapted to teosintes. As maize developed and evolved, was selected for more maize-like characters, those dabulous leafhoppers moved out into these new types of plants.
Lapel up.
Sure.
Right here.
What, I'm not talking loud enough?
No, you're doing great. It slipped on the thing I was trying to.
Oh.
So.
Should I stand still?
Nope, absolutely not. That's why.
I'm not very good at standing still. Okay, good. All right, so as the dabulous leafhoppers evolved, they moved into these new ecosystems, and that's how we got the modern dabulous species that we have today. All right, so here is a progression of maize as it evolved, the early teosintes, and then this is what the early Mesoamericans had down in central Mexico, down in South America, and into North America, eventually coming up with modern day maize. So here's a progression of the leafhoppers. They, most of them still will feed on those early progenitors, some teosintes, some gamma grasses. There's some that specialize in Tripsicum and maize. There are two species that are fully adapted to life on maize. They're maize specialists. Those are Dabulus matus and Dabulus elamatus. And what these structures are, these are the male genitalia. This is how we distinguish different species by pulling the male genitalia, and you can see that they differ in their structure, and that's how we know which species are which. Okay, so here's a big question. Where did Spiroplasma cunculi come from? I'll start off by saying that spiroplasmas, as a group of bacteria, are found in many insect orders, the flies, the bees and wasps, the hemipterans like aphids and leafhoppers, odonates, Lepidoptera, and beetles, etc. Corn stunt spiroplasma infects two hosts, leafhoppers and maize, a plant host and a leafhopper host. Corn stunt spiroplasma causes disease in its maize host. It's lethal. It will kill it, but not in its leafhopper host. It's slightly pathogenic, but it doesn't kill the host. This suggests a much longer association with leafhoppers than it does with maize. Therefore, it's thought that corn stunt spiroplasma first infected leafhoppers and became endemic, if you will, within its leafhopper host, and then moved secondarily to plants. And this happened when maize was selected for, when it was domesticated in Mexico. So where did corn stunt come from? It came from the hoppers. Okay, so let's go back to early history and when it was first found in the US. The first record that I could find was a 1946 paper by Kunkel, a leafhopper transmission of corn stunt. And he was describing the 1945 outbreak in Texas and California. And Kunkeli is named after this gentleman. Okay, biology of Spiroplasma kunkeli. After this first description, it was initially thought to be caused by a virus because they didn't know about plant pathogenic bacteria in the phloem at that time. Bob Davis and his group isolated, captured images back in 1973. This is disputed because I just found a paper by Bob Granados describing the same kind of morphology in 1968. So this is a little bit questionable to me. Okay, so spiroplasmas are prokaryotes in the class Mollicutes. This means thin skin, Mollicutes. These actually inhabit the phloem of plants. This is the only place that they can live. These plant pathogenic spiroplasmas, other Mollicutes, they infect humans, animals, whatever, but this particular group are phloem infecting pathogens. They have no cell wall, they're pleomorphic. So most bacteria, have a cell wall, these do not. So they can move, they can change their form. They inhabit the maize phloem. They also inhabit insect hemolymph and their tissues. So they infect the whole insect, but they don't infect the entire plant. And they propagate in both hosts. Okay, so the interesting thing about spiroplasmas, they're cultivable. This is amazing. Most phytoplasmas are not cultivable. So spiroplasmas serve as a model system for phytoplasmas. And we have here at Oklahoma State, we've got isolates from the US, from Central America, and from South America. So we have a very nice culture collection we have available to us to study. All right, so let's move on to the vector. The most important vector in this system is Dalbulus matus. This is the corn leafhopper. And this is a nice big picture, so I'm going to point out the importance of the two eye spots, tan color, that variable, and kind of bullet-shaped animal shown here. Nice spines on the metathoracic legs, identifies it as a leafhopper. Okay, so identification, I told you, is through the male genitalia. So what we do is we whack off bug butts and we clear the tissue, and then we look for those structures, the style here, and these can then be used to do phylogenetic analysis. Nowadays, we do sequencing. It's much easier and faster, but if you're out in the field, you don't have access to at least I don't think you do. You might, because that technology is becoming available, and you don't have time to clear the tissue. So there's other characters that you can use to identify Dalbulus matus. So this species is a small yellow to tan leafhopper, 3 to 4 millimeters in length. And also, I forgot to mention, I have some handouts that show some of these hoppers that I'm going to discuss. If you want one, you're welcome to come and pick one up later that has these. What I want you to notice are the very prominent black spots on the vertex or the point of the head. And this is important. The spots are surrounded by a white halo. I hope you can see this. And then if you look at the face head on like this, you'll notice that there's nothing else. You just see those two dark prominent eye spots. And that helps to distinguish it from its congener, from its fellow genus member, Dalbulus elamatus. And I'll show those to you next. So I've got other small yellowish leafhoppers in the maize ecosystem. The first one is Dalbulus elamatus. It's a congener to Dalbulus matus, but it's distinguished by, it has the two dark spots above on the vertex, but it also has two small spots just below that. So it has 4 spots. Dalbulus matus has 2 spots. Okay, that's how you tell. And almost doesn't make any difference because this one is also a very efficient vector of corn stunt spiroplasma. The other leafhopper I want to draw your attention to is Cicadulina umbella, the African corn leafhopper. We do not yet have this in the United States, and I hope we never do. It's in Africa and parts of Asia. It's a very bad leafhopper. If you think you see it, please call USDA APHIS right away. Okay, we don't want it because it transmits maize streak mastery virus, a devastating maize virus. We do not want it in the United States. All right, other hoppers that you might find in the maize ecosystem. This one is Balclutha neglecta. You see how closely it resembles Dalbulus matus? It's a small, it has kind of tan, but it lacks those spots. Here's another one, Gramenella ficii. This is a Gramenella species. It's the same color, but it has 4 lovely spots on the fronds of the insect. And another one, Planocephalus flavocostus. There's lots of little yellow leafhoppers in maize and grasslands adjacent to maize, and so it could be confusing. So focus on those yellow spots. All right, I want to talk a little bit about misidentification. You guys, as crop consultants, you may be called out and you see these hoppers, you'll go to the internet to try to get an ID. Please be aware that there are websites that have incorrect information. I just want to show you a couple of examples. Here, I'm sorry? Don't get me started. Yes, there is. It's important to go to a source that you can trust. These are just a couple examples. This website from South America, this is about Clutha species. It's not Dalbulus. And here's one that identifies Dalbulus natus, but that's clearly not even a leaf hopper. This is a plant hopper. And then here's two images. These are Dalbulus. I've seen them, but they've been colorized. They're green in these images. Dalbulus is not green. They don't have any green coloration whatsoever. And so it can be misleading. You might see, oh, there's a slightly greenish leafhopper. It has spots. Is it Dalbulus? Well, the answer is no, it's not, because Dalbulus is yellow to tan, no green. How do I know this? Because of this image. Thank you. Yeah, technology. This is Dalbulus matus for sure. How do I know this? Because I was there in 1989 when we took the insects out of colony to take these photographs. So any picture that if you go to this one, you'll always get the right answer, okay? Okay, other important maize pathogen vectors. There's several, but I want to mention these four. The Gramenella nigrifrons, this is the blackface leafhopper. It's the vector of MCDV, and also it's an experimental vector of corn stunt. Macroceles quadrilineatus, this is the aster yellows vector. We have aster yellows seriously in the state of Oklahoma. You'll also see it in the maize ecosystem. In fact, it will transmit aster yellows to corn. And it's often thought of it, we often will dig it up when we're assaying for maize stunting pathogens. We'll see aster yellows. Euclideus variegatus, big, huge, robust leafhopper. It also occasionally shows up in corn. And then finally, Exidianus exidiosus. This is the gray lawn leafhopper, mostly in Bermuda grass and other grasses, but it will show up in the corn maize ecosystem. Important because it's also an experimental vector of CSS. In fact, there's a lot of experimental vectors, meaning that they were forced to transmit corn stunt in the laboratory. How important they are out in the field, we do not yet know, but we're hoping that our colleagues will be able to answer those questions. Okay, Dalbulus matus, life characteristics. Let's go through these pretty quickly. First of all, Dalbulus matus is aggregative. If you've seen this in the field, how many of you have seen Dalbulus in the field? Oh, we have a few people, good. In the back. If you look in the world, you'll see them if they're there. And you'll see that they're aggregative. Why? Because it's protected, they lay their eggs in groups, and they lay a lot of eggs. This is a nice photo because it shows two color morphs. I just want to point this out. Here's a lighter tan color, yellowish form. Here's an older leafhopper. The older they get, the darker they get. Okay, so bright yellow to tan, darker tan. Okay, Dalbulus exhibits exponential growth. That means one hopper who's fertilized, if it's a female, can lay some eggs, and then that population will grow rapidly. Not as fast as aphids, because those are clonal, but they will replicate and reproduce very quickly. Egg oviposition to adult takes 25 to 35 days per generation. They lay eggs in clusters of 4 to 10. That number changes depending upon who you talk to. A single female can lay several batches of eggs throughout her lifetime, and that lifetime might be several months. Okay? In the literature, estimated anywhere from 2 to 12 generations per year. Again, highly variable. There's, I saw one report, 14 generations in the state of Arizona. Of course, they have continuous corn. So they're going to keep reproducing as long as they can. All right. I don't know how many generations we might expect to see in the state of Oklahoma, but that's going to depend on conditions at the time. All right, reproductive hosts of Dalbulus matus. I found this nice table, and basically maize or maize relatives only. They don't reproduce on other grasses. Reproductive hosts are only maize. That's important. However, they feed or can overwinter on a number of different hosts, at least according to the literature, and some of my own work. Okay, in Mexico, it's maize and the teosinte and other grasses where they are known to overwinter. In the US, it's suspected that Johnson grass, sorghum, wheat, oats, alfalfa, the gamma grasses, panicums, volunteer maize, Bermuda grasses, other dicots. I happen to know personally that Johnson grass, oats are excellent feeding hosts for Dabulus matus. I did this work as a graduate student. They probably feed very well on some of these other hosts, but again, they do not reproduce on those hosts. Here's an important little factoid. Very low numbers of Dabulus matus were shown to survive in Mexico in the complete absence of maize for several months. We're talking like 5 to 7 months. So without maize, they can still survive. And then I found a work by a fellow graduate student of mine. Yeah, he starved them for 2 months on wet sand. They lived for 2 months. They can live without food for up to 2 months if they have moisture. So these guys are extremely resilient in their ability to survive. How am I doing, Ashley? Oh, I'm good. Thank you.
But someone had a comment back here.
Oh, yes. No, they cannot. It takes me 24 to 36 hours to kill them when I tuck them in my -20 freezer. Okay. Yeah, I think Ashley's going to talk about temperature. Yeah, that's a very important concept to be hanging on to. All right. Here's another one. Dabulus matus, like other supervectors, is migratory. Migratory. Okay, so historically, they overwinter in the highlands of Mexico. And there's El Norte winds that help with local movement within that country, move them away from fields and then back into fields. Okay, these winds, if they're strong enough, can result in what are called exodus flights. These are the long distance flights into other parts of the country or even other regions of the continent. And those are the ones that we are concerned about in the state of Oklahoma here. Where do they go? Sometimes they're captured further north or south, historically in California, Texas, and other Gulf Coast states. Okay, that really wasn't us until recently. All right. So Dabulus is migratory. What triggers those massive exodus flights that occur occasionally? So this was a great paper by Taylor where they analyzed different environmental conditions, and they found that when insects were reared under late autumn conditions, like a short photoperiod, lower nighttime temperatures, and senescence of corn, that's when they would take off. And if those strong winds are present, that can be used to carry them further and further away. They have to be very strong because Dabulus is not considered to be a strong flyer. It's almost a passive flyer, relying upon those winds to carry it long distance. Okay, so everybody always asks, well, what about hurricanes and storms? Yes, they're very likely to play a role. I checked the dates of hurricanes this year, and Alberto occurred in June. I'm thinking, wait, that's too late because we had corn stunt here in Oklahoma in June. So I'm not going to talk about that. I have ideas, but I think it's a little bit premature. But I just want you to know that there are short hops, if you will, short migration, and then long migration. And maybe the way that they got to Oklahoma and further north were through a series of short hops and then longer migration events. I don't know what they were. Okay. Transmission biology. Corn stunt is acquired during feeding from the maize phloem. The cells, the spiroplasmas, are ingested. They're taken up by the gut. In the gut tissue, they replicate, they multiply to pretty high numbers, and then they break into the hemocoel. That's the open circulatory system of insects. Within the hemolymph, they migrate to the salivary glands, shown here, where they invade the salivary glands. And then again, they replicate to high numbers, at which some point they make their way to the salivary ducts, and then they can escape the insect. There's only one way in and one way out, and that's through the gut and out through the salivary glands. They don't poop it out the back end, anything. They have to come from the phloem and go back into the phloem of plants. So that's the only way. All right, so these are propagative. They have to multiply in their vector in order for the vector to become inoculative. And it takes a long time, 3 to 5 weeks from acquisition, from culture or from the plant to inoculation. It's not rapid like with many aphid transmitted viruses. It takes a long time. Okay, these are just some pictures that I took years ago. Diabolus matus, looking at, here's the salivary gland structure pulled out. And I just want to show you that the complex of the salivary glands, it's very complicated. So there's all these ducts. The spiroplasmas have to make their way to the forming salivary duct. And we hit the money shot. We captured an image of the salivary duct with an opening. And this is the pathway that it takes to get out of the insect. And then later we were able to confirm spiroplasmas within their insect vectors. So this is an image within the salivary gland of a pleomorphic spiroplasma. They're not helical in the insect. They lose helicity. Here's one of them invading the salivary glands. These are spiroplasmas. So it's just kind of an interesting thing to demonstrate this. All right, the last thing about features, I want to mention feeding behavior. Oh, I really want to talk about that, but we don't have 3 hours. I've got 3 minutes. So I've done a lot of work looking at feeding behavior of Diabolus matus. And the question is, why is it so efficient at phloem-restricted pathogen transmission? The answer is because it's primarily a phloem feeder. And so I and my students have recorded Diabolus matus using a technology called electropenetrography. Briefly, we wire up insects. We glue a gold wire to their pronotum, and then we place them on an electrified plant. When the insect probes, it completes a circuit, and then that insect acts as a variable resistor. And the output is a bunch of squiggly lines, which are then interpreted to represent specific behaviors. I'm not going to go through the whole spiel, but I'm just going to tell you, I'll show you here. So that S-shaped curve, that's initiation of phloem probing. The long straight line is phloem ingestion. Then she farted around a little bit, did it again, long phloem ingestion. Then some xylem ingestion here, farted around. Sorry, shouldn't use that word. Goofed off. Then she initiated phloem ingestion, and here she fed for a very long time. This is a 24-hour sequence, 1 day. She spent most of her time feeding from the phloem. The vast majority of Diabolus hoppers do the same thing. They are really good at tapping into and ingesting from the phloem. Okay, this is the primary reason why we get almost 100% transmission using single Diabolus matus leafhoppers when they've acquired corn stunt spiroplasma. Extremely efficient. That's our problem. All right. Oh, I just have to show this off. This is the phloem ingestion waveform when it's expanded out so that you can see the detail. This is the sucking pump picking up, oh, it's sucking from phloem and sucking from phloem. And they'll do this for hours and hours at a time. It's so cool. All right, enough of self-promotion. All right, history of incursions in the US. People ask, how long has this been here? Is this the first time it's come? The answer is no. This isn't a new disease. It's a re-emergent disease within the United States. And it's probably been here for generations, but not all the time. Coming and going, because it hasn't overwintered in the States until recently that we know of. The first report of corn stunt was a 1942 outbreak in California. And then I think he was talking about a similar outbreak in Texas. You'll notice that these two papers were in the same journal. Here, this one was an earlier, i.e. earlier in the publication than this one, but they occurred at the same time. So I don't know who gets the credit. Then there's another mention of an outbreak in 1964. Mira, she was a very famous vector entomologist in phytopathology. No subsequent paper, just an abstract. The next mention is an incursion of Dabulus matus into California without corn stunt, as far as I could tell, in 74. And then the 1979-1980 outbreak, this was a major outbreak of corn stunt and Dabulus, California, Texas, and Florida. This is when Jim Tsai at Florida, I think he was ARS, actually isolated the pathogen, and that's the source of our Florida isolate that we have in the freezer here. All right, then in the 1980s, there were several reports of Dabulus that were likely endemic in California at the time. This is Charlie Summers' paper where he discussed overwintering hosts, okay? So it's probably been endemic in California since at least the early 90s, probably before that. So that means continuously living in the state of California. There was a 2016 incursion of Dabulus in Texas in the Rio Grande region, documented, but without the documentation of corn stunt, as far as I know. And then finally, our outbreak here in 2024, which was major, and you guys are going to talk about that. Okay. All right, so where are we now? Before domestication of maize, corn stunt, and Dabulus were restricted to Mexico, central Mexico. After domestication of maize, we have a distribution throughout the Americas. This is an incomplete map, I'm quite sure, but in terms of documentation, you have to wait for that to come out. I think you guys are going to update, maybe? Okay, good, thank you. So we have a pretty serious situation, and our responsibility in Oklahoma, we're placed very importantly with respect to this disease. I'm going to stop now, and I got it wrong. I thought Ashley was going to talk, but I'm going to go with Dr. Duffick. So she's going to tell you what's happening with respect to the pathogen itself. Thank you.
And we'll do questions at the end for all three of us. Oh.
Can you guys hear me?
If you have a question for Astrid, real quick, while that's getting loaded up, let's do that. Go ahead. Yeah.
So those outbreaks through time, how long live for?
Yeah, 2 years.
This
So,
is hers.
oh, thank you. As far as I can tell, 1 year events, except for that one in 79 and 80, and they probably overwintered in Florida and maybe in other regions. I just don't have access to that information. Probably a mild winter, so they were able to survive.
Where should
All right.
you are moving or I'm moving? All right. All right. All right. Good morning, everyone. My name is Maíra Dufek, and I am the row crops extension pathologist at Oklahoma State University. So thank you, Astrid, for this beautiful introduction about corn stunt and corn leaf hopper. But as you guys can see, she's more like a lab rat, and I am more like a field rep. So I'm going to show a lot of pictures here. So I'm here today to talk about this new disease in corn that's called corn stunt. And as you guys already saw, based on Dr. Anda, the presentation, that is not kind of a new disease. We're calling a new disease, but was already present in the United States a long time ago. The problem is that we didn't see this disease for years, for almost 40 years, and we ended up forgetting about it. I just would like to highlight here that Oklahoma State University was the first university in the United States that in 2024 brought the attention for growers and stakeholders about this problem that we are having in corn production in the United States. So I remember receiving my first call about corn stunt from a grower. He said, I kind of have a viruses or a bacteria disease on my field, and then I stood my feet with my crew. Part of my crew is here today with me, and on August 1st. And then on August 1st, we entered the field and we saw this cloud of insects moving around. And I am from Brazil, not sure it's a good thing or not, but we have been dealing with corn stunt for a while already. Since 2015, it's causing us a lot of trouble. And I remember saying this out loud, like, this is Daubulus made, is like, this is cigarrinha, that's the name that we say in Brazil. And I remember my heartbreak kind of increased a lot and started beating fast, because I was like, it's not possible that's here already, because we know how damaged this disease can be. And I was in the first time, like, scary when I saw this in the field. So the first real report that we made it for growers and stakeholders was in August 12. Took us a while to really confirm the presence of this disease in the state, because as I'm going to show throughout my presentation, we have a wide variety of symptoms that this disease can cause. And just by seeing the symptoms in the field is not able for us to really identify and say, okay, we have corn stunt. We need to send samples to the lab and identify at least one of the pathogens that are involved with this complex that we call corn stunt. So here's the first report. If you guys wanted to scan this QR code, you're able to see the information about corn leaf hopper here in Oklahoma when it was first published. And we have also a good summary about this disease also. So the first report of corn stunt here in the state of Oklahoma, as I mentioned, was in August 1st. It was the first time that we step our fields inside a corn field that was showing symptoms of corn stunt. And then I was like, this is not a problem. I'm a plant pathologist. I work with plant disease, but this is not a problem that a plant pathologist can solve by itself. I need an entomologist here. So I call Ashley, and Ashley was already kind of aware of this problem, because she received some calls about it too. And then we contact Dr. Austri to help us. And because she'll have been working with diseases for 30 years already, so she knows how to identify especially the corn leaf hopper, the insect, and also the symptoms that are associated with this disease. So the location was in Grady County. The first field was a full season corn. The type was grain corn. And then the problem with that is that when we received this call, the corn was already in the senescence growth stages. So it was very difficult for us to really say, okay, this is corn stunt, and the damage that we're seeing in the field is caused by this disease, or this is just like drought, stress, or other factors that can affect also the corn crop. And we also was difficult for us to not mistake those symptoms with just the senescence of the crop, right? So it's easy for us to identify corn stunt when we are in the beginning of the reproductive stages. Then later in the season, we start to miss some of that information about the symptoms. And after Oklahoma State University published the first report, we kind of stood up. We were brave enough to say that, okay, we have a problem in the state. May other states have the same problem, and we know what it is. And we are the first one that kind of named that. We named corn stunt. Maybe other people, other extension personnel in other states have been seeing that, but they are not really aware of what was it, or they are not really brave enough to say the word corn stunt. But after that, we had several states that follow us. So as you may see here, we have the report in Missouri, New York State. I'm not sure if you guys know about that, but this disease was also present very far east, northeast this year. We have South Dakota, Minnesota, Kansas, and also Texas. So after report corn stunt, other states kind of was aware of the problem and start to also go to the field, scout, and send samples for disease identification. But what causes corn stunt, right? So Austria already mentioned that corn stunt is caused by a complex of 4 pathogens, right? So just to highlight again, we have 2 bacteria that are involved with corn stunt and 2 viruses that are involved with corn stunt. So you can imagine if we have a disease in the field that is just caused by 1 pathogen, sometimes it's already very hard to manage, right? So imagine us to manage a disease that's caused by at least that we know so far 4 pathogens involved in this complex. But in the United States, we just report so far 3 of those pathogens. So these 3 pathogens, 2 bacteria, and maize hyadophinoviruses was already reported in the United States in this growing season or in previous growing seasons. But maize straight mosaic virus was not reported yet, was reported in Brazil associated with this complex, but was not identified here in the United States so far. What are the corn stunt symptoms, right? So I also already showed some symptoms, but I'm gonna go a little bit more deep into it. And I expect that you guys, in the end of this presentation, be able to next season, if you see something similar with that, give us a call and send us samples to confirm if it's corn stunt or not. So I have all these pictures that I'm gonna show for you guys today. They are taken from Oklahoma. So it's from here, from our state. So I'm gonna have several cases in here. So I have first pictures of full season corn that was planted early in the season. And then when we arrive in the field, we mix kind of those symptoms with senescence already. But the most common symptom that we saw here in Oklahoma in the full season corn or in the double crop corn was this reddening of the leaf tips. So as Austria already mentioned, these pathogens, they move inside of the phloem of the plant. So they tend to concentrate in the organs, the parts of the plant that are growing. So that's the reason we tend to see the symptoms of corn stunt, these reddening strips in the upper part of the plant first instead of the lower part of the plant. So we tend to see this reddening in the upper part of the plant. Here's one of the symptoms. Another very common symptom of corn stunt when the plant is infected early in the growing season is this shortening of the internodes, as you may see here. And this is causing the stunt of the plant, right? The plant is getting stunted. And that's the reason this disease is called corn stunt, actually, is because of this particular symptom that is the shortening of the internodes. Another symptom that we observed here in the United States, but also is very common in other countries such as Brazil or Argentina, they already mentioned that, is that stay green effect. So especially in the borders of the field, we start to walk in the border, you see that the field, the plants are not affected. They are already in the senescence growth stages, already getting dry. But the plants that are very infected with the pathogens, they stay green. What's the reason that you think that they stay green? Some people say that the pathogens manipulate the growth of the plant. But what we have been noticed is that that plant stay green because it never reached the reproductive stages. So you can see here that there's no ear being formed here in none of these plants. And we tend to notice that these plants stay green for longer because never kind of passes that reproductive stage and then kind of have this effect of staying green until the end of the season. Another symptom that we also see is this chlorosis streaks. I didn't observe this a lot in grain corn here in the state. I observed more in sweet corn, but we also, this is grain corn, and we also observe this chlorosis of the leaves caused especially associated with Spiroplasma conchyli. Another characteristic that when you go to the field, actually when I received one of the calls, I received 2 calls about corn stunt before I knew that the disease was corn stunt. So a grower called me and he said, I have a problem with my corn field. And every time that I go inside field, I come back with my clothes full of a black moldy. And then my first thought was actually gauze wilt, because we have this sometimes in the state. And then I was like, maybe it's a bacteria disease. And then after that, I understood why he told me that his clothes were turning black when he used to go to the field and scout. Because actually the insect vector that transmits those pathogens, the cornleaf hopper, when he's feeding on the plants, he expels, and then we have the production of this black sooty mold, this honeydew that accumulates on top of the leaves and also in the upper part and the lower part of the leaves. So when we have a higher population of the cornleaf hopper, this is very common to see in the plants. this production of this black sooty mold that sometimes Ashley's going to talk more about the insect part, and she's going to mention that the insect is very small. So sometimes we miss the insect because they move fast, they hide into the plant. But then if you see this kind of symptom here, it may be because the population of the corn leafhoppers is very high into the fields. Another characteristic that we have, and as I said, corn stunt caused so many symptoms that sometimes it's difficult to separate what was caused by drought stress in the state. That's also very common. What is a nutritional deficiency, such as potassium, that also can cause this reddening of the leaves of the corn plant. But here we have another symptom that is missing kernels or light kernels. So here is some comparison of the years of a plant that was looking healthy and a one that was looking infected. So the kernels doesn't finish to fill, the plant doesn't finish to fill it up the kernels. We have here, especially in the tip of that plant, that kernel, and also here we see the difference in size. So this is going to affect the yield because the grain is going to be lighter or they're not going to be produced. And we also have this other effect that they call loose teeth. So when we look in when we grab a corn ear, we can see that grains are not very attached to the cob. So it's very easy for us to pull up that grains from the cob. And then here we have a comparison of a health. But what I'm saying health here is because sometimes the plant have the pathogens inside, but was not affected in the way that another plant was, right? And here was what I'm calling like the infected plant. You can see the difference in the size of those grains, and they are light in weight. So it's going to affect the yields, of course, in the end of the season. Okay, so these are the symptoms for the full season corn. But what did we see for the double crop corn? So Oklahoma is a state that has this unique crop system where we can also have the corn that's planted early in the season, but also after harvesting wheat or late in the season is an option for the growers too. So we start to monitor the fields as early as we could. Most of them are already like almost in the reproductive stages. But then we start to see these beautiful symptoms here that for plant pathologists is beautiful at least, right? These reddening streaks in the leaves, this chlorosis also in the leaves, and the production here in the beginning of that black moldy in the plants. Here again, this is a field I was lucky enough that Dr. Josh Lofton works with this double crop system here in the state of Oklahoma. He has some fields across the state that he planted late, and I was able to monitor more the symptoms closely in that fields. So here's an example of a field here in Stillwater that was planted late in the season, and we can see this very beautiful reddening of the leaf tips. For the corn plants, we can also see this example of a completely stunted corn. And what I'd like to highlight here is that all the pictures that I'm showing is from fields from where we saw the symptoms of the disease, but we also brought the samples back to the lab and we characterize at least one of the pathogens involved with this complex. So we are sure that we have the presence of the pathogens in those fields. Another symptom that we observed a lot in the late planted corn was this production of multiple ears. So as we see here, when you say production of multiple ears, we tend to see that's good things, right? Because we're going to have more yield if you have more ears in a corn plant, but that's not the case. So the ears are going to be produced very small, multiple in a corn plant, and they're not going to fill it up they're not going to finish to fill it up the grains. So as you may see here in the first plant, we have one ear being formed here, another 1, 3, 4. So here's another example. We have 1, 2, 3. I think there's another one here that I'm covering. Here in another example in sweet corn, very distinctive symptoms in a sweet corn plant where we have 1 ear, 2, 3 being formed, and none of them is going to produce or finish to fill it up the grains. So that's the problem with that, because we have several. What we understand about the plant is that the plant's kind of trying to fight against it and then produce 1 ear and say this one is not going to be enough. Produce another one, and then ended up that none of them the plant can really finish to fill it up the grains. So here's an example of a plant that since from the ground already we have the production of the ears, and we have 6 ears in a plant. Here's a Another example of a plant that also is a sweet corn that also have 6 ears being formed. And since the ground here, we can see the production of this ear already. So it's changing a lot the physiology of the plant, the multiplication of those pathogens inside of the corn. In sweet corn, as I said, we have different kinds of symptoms. So you start to see this in the beginning of the symptoms expression. The sclerosis here, very light symptoms, but then they become like this reddening streaks, and also the sclerosis, very distinctive in sweet corn. And you see that they're very affected because sweet corn is already a short plant, but when it's infected with corn stunt, it becomes even shorter. But is corn stunt a new disease? So I also already mentioned this. Actually, the first description that they consider like the original description was in Rio Grande Valley in 1945. And after that, we saw some reports of this disease causing economic damage, especially in Florida. I think there is after Hurricane Davis, 1960 something, they report the economic losses in there. So they have like a real report of the economic loss being caused by corn stunt. But and then this disease caused some damage from 1945 to 1980s, especially in the South Texas, Florida, California, the states that are more warmer, that allow the corn leaf hopper to survive throughout the winter or other conditions. But then we have this kind of period that no one talk about corn stunt anymore, neither the corn leaf hopper. So for 35 years, basically, we didn't see the presence of this disease, neither the insect vector, and we kind of forgot that this could be a problem for us. So in the South America, we start to see the reports of this disease in 2015 again, in Brazil, Argentina, really hit Argentina crop production in the last year. But I think we're too distracted with other problems and we kind of forgot that this would be a problem. But we have people like Austria that have been working with that for more than 30 years because she was like, this is going to become a problem in the United States. I just don't know when, but that eventually will. So we're very lucky to have her around. She's retired, but she's working more than we are working right now. I don't think she knows how retirement works, but she was a great source of information for us because we're able to really confirm that everything that we're seeing was true. And then in 2016, they saw a reemergence of the corn leaf hopper in Texas, but they didn't see a lot of symptoms of corn stunt or yield losses associated with that disease. And now in 2024, we saw this large expansion of the corn leaf hopper and the disease from the south of the United States toward other states. Here's just another example of a report that I was able to find from 1964 from Mississippi State University. And here they didn't have the confirmation of the pathogens that are inside the corn plant. But they did this report and this map based on the symptoms that they're seeing in the field. So extension personnel, such as myself, that work a lot in the field, they said, hey, these symptoms are looking like corn stunt. And then they drew this map. So you can see here in the southern United States, a lot of states being, they marked here in black the counties that are affected. But you can see the disease was even reported far north as Ohio. So it was already present in regions that we consider that to be more cold, and the probability of having this disease there was lower, right? But was still reported as far as north of Ohio. So when we receive this call about corn stunt, we start to see the symptoms across the state. We set up what we call this corn stunt team here at Oklahoma State University. So we had a specialist that worked for 30 years with that. Dr. Ferris just joined the team, but she was able to help me to set up everything that was needed to confirm the diagnosis of the insect vector for her side. And then here we set up what we call this corn stunt team. That was Jane Onso and Sarah Wallace from the Plant Disease Diagnostic Lab. They are the ones that are confirming the identity of the pathogens involved with this disease. Dr. Spindola, he works with the sequences, so everything we need to publish the sequence to confirm in a database that we call GenBank. So he was doing this for us because he works with more the molecular side of things. I don't work a lot with the molecular part. I'm trying to establish this on my lab, but as I mentioned, I'm more like in the field side and the extension side. So this is our team that we set up, and then we start to we start with this team be able to identify the pathogen that was associated with this disease to really provide a confirmation and bring awareness of this problem. So here's a map of the distribution of corn stunt in 2024 growing season. And all those counties here that are marked in this map, they are reports that were confirmed here from Oklahoma State University. So all the other states send samples to us. We serve kind of a national hub for receiving these corn stunt samples. And we are able to provide confirmation of counties, fields that are having corn stunt symptoms in all those states, including 6 counties in Oklahoma, 14 counties in Kansas, 1 county in Nebraska, 1 in South Dakota, 1 in Minnesota, Wisconsin, Indiana, Missouri, Arkansas, a very down south one here in Alabama, and also in New York State, we have 4 counties that were confirmed with corn stunt pathogens, especially focused on the Spiroplasma conchyli, as also already mentioned, that's the main pathogen involved in this complex. Why we were able to do this so fast compared with other states? Because actually, also already mentioned, we had a collection of Spiroplasma conchyli here available to us. So we have what we call, when we are doing research, that's our positive control. So we kind of have this gold mine here that was able to us to have this positive control and then sequence what was generated by that amplification to really confirm that that was Spiroplasma. And as I mentioned here, we start the first report with Oklahoma in August 1st. But then the last one was Alabama that just sent the samples 2 weeks ago. So we're able to confirm because they still have some corn in the ground. All the other states didn't have any more, unfortunately, for us to test. We know that Illinois also have corn stunt, but they are still working the first report. They didn't join us. And also Louisiana, we believe that they also have, but they have a trap that actually can mention more about it and was not functioned this year. So they're not really aware of the presence of the insect, but they are also, let's say, join us for the efforts for other years in order to identify this disease. And also you're probably wondering, where is Texas in this map, right? Texas is missing out here. So here we have a map that actually we pull out from a presentation from a webinar that was conducted by Pioneer, that was Understand Corn Stunt. And this was reported that actually Corteva, they have a diagnostic lab and they are able to collect samples across these states and confirm the presence of corn stunt. So in Texas, we have a lot of reports of corn stunt, also Oklahoma, Kansas, Missouri, and New Mexico. I'm still learning United States geography, so of course, the Texas also joined the map of corn stunt in the United States in 2024. How does the corn leaf hopper transmit the corn stunt pathogens? So she also mentioned a little bit about that, but I'm going to go a little bit more deeper in that. So we have here a diagram, a figure showing how the transmission works. So here, a lot of people already studied this pathosystem in other countries because this has been a problem for a long time in other South American and Central America countries, excuse me. And then here we have in laboratory conditions, they did this experiment and they know exactly how long it takes for the corn leaf hopper to feed in a corn plant that is infected of those pathogens and then be able to transmit those pathogens to other health plants. So here we have when you see CSS in the in this image, it's referred to corn stunt spiroplasma. When you see MBSP is maize bush stunt phytoplasma, that's another bacteria. And then you see these letters here, MRFV is referred to maize hyadophinovirus. So let's start here in number 1. So we have the corn leaf hopper that is feeding into the phloem of an infected plant. And then for acquiring that pathogens from the phloem of an infected plant, for the spiroplasma needs 1 hour of feeding. For the phytoplasma needs 2 hours, and for acquiring the viruses needs 6 hours of feeding of that insect in an infected corn plant. But as I also already said, this insect feeds a lot, right? So most of the day, this insect is just sitting there calmly in a spot that he found comfortable feeding in a corn plant. So in 1 day or less than 1 day or in maximum of 6 hours, all these pathogens can be acquired by the corn leaf hopper and start to multiply inside of this insect. So here in number 2, the pathogen here is just a representation, is multiplying inside of the hemolymph and establish is the circulation inside of the leaf hopper. But then this leaf hopper cannot transmit right away those pathogens to a health corn plant. So it needs to replicate, and this takes around, here we have the days, 17 to 23 days for the spiroplasma, 22 to 20 days for the phytoplasma, and then for the viruses around 8 to 22 days. So it's to say that around 20 days, these pathogens needs to be inside of the adult, the corn leaf hopper, multiplying to be able to then this corn leaf hopper to start to transmit for a healthy plant. So it's not right away. It's not that it's going to acquire today, it's going to transmit tomorrow. No, it needs to have some time. And then the transmission of these pathogens occur in a shorter period of time, actually, that needs to acquire. So to transmit for the spiroplasma, it needs to be 1 hour of feeding again in a health corn plant. For the phytoplasma, it's just half hour is already enough to transmit inside of the phloem of the plant. And for the virus, that need around 8 hours. So in 1 day of feeding in a plant, that plant can be already infected with those pathogens. So when this corn plant can be infected? So unfortunately, the corn plant can be infected from after emergency through almost like senescence. So the corn plant is susceptible throughout the whole growing season. However, what we know is that when the plant is infected here, in the vegetative growth stage is when is the critical period of time for us. Because if the plant is infected here in the vegetative growth stage, those pathogens is going to have time to multiply inside of the corn plant in a way that's going to affect here the reproductive states and our yield, the productivity of those corn plants. So here's the critical time that we need to prevent the infection. And I'm going to let Ashley talk more about this. But here's when the plant is going to get infected, but the symptoms of that disease we're going to see just in the reproductive stages, because we need some time of those pathogens to multiply inside of the corn plant. We used to say 3-4 weeks for us to be able to see that symptoms in the field. So when I received that call about corn stunt, we confirmed corn stunt on August 12, based on the lab confirmation of the identification of the pathogens. I was like, I need to do something, but I didn't know exactly what, right? So we have this field here in Stillwater. I took a picture by drone. That was also confirmed that we have corn stunt in this field. And then I asked the permission for Dr. Josh Lofton, because he's the one that owned that corn field. Actually, in the end, I was owning this more field than him, but he kind of donated for me. And what I did was like, I would like to understand the distribution of the symptoms. When we're going to start to see the symptoms in the field, and how will be the distribution of those symptoms inside of this field. So this is about 1 acre, it was around 25,000 plants. And what I did was kind of crazy. I have some people that work in my lab. So every week I told them, you need to go to that field. You need to count plant by plant, and you need to stop each one of them and see if they have symptoms of corn stunt. And they did it. 3 of them, they spent like 1 whole day in a week doing that. We have cases of a student that hurt the eye with a corn plant, ended up in the emergency room, and we have all cases regarding this project. But it was a fun experience, and they did it. And when at the end of this project, I see that I have 25,000 plants, and they count one by one. I was like, wow, I was crazy. And they are more crazy that they accept the challenge. But this provided us some nice information. So when I say what symptoms, so I asked them, go to the field and look for those symptoms. Production of multiple ears, reddening of the leaf tips, this kind of reddening streaks, stunt of the plants. And also something that I didn't mention here, but also when the plant doesn't have any ear, doesn't produce any reproductive parts of the plant, was also one of the symptoms. And they are able to do that for 4 weeks before the plant starts to senescence, and then we are not able to differentiate what was corn stunt, what was senescence. So here we start on, we confirm corn stunt on August 12, And then on August 15 was the first time that they started doing this study, August 22, 3rd, and also September 6th. So here's the result for the first week. So in green here, we have the disease plants, the plants that are showing symptoms of corn stunt, and in black, we have broken plants, just plants that we lose in the field due to other factors. So as you may see here, in the first week, we already have the presence of some plants, especially here in the border, in the middle, that are showing symptoms of corn stunt, and also in the border here. In the second week is what we saw. We start to see the increase of that diseases plant. In the field, especially in the border, we have this border effect. What makes sense because we imagine that the corn leaf hopper is migrating through other fields to inside the corn plant, and then they stop at the borders, and then they migrate for the inside part of the field, right? This is the result for the third week. We're like, okay, this is increasing. We don't know how much, but this is increasing. So we can see a lot of green being marked here, showing that they have the symptoms of the disease. And this is our fourth week. So in the fourth week, you can see that almost all the field was showing symptoms of corn stunt. And I need to mention here that in this case, we didn't use any measure to try to stop the reproduction, the growth of the Dalbulus matus, the corn leaf hopper. We just let them there. We know that they're there, we're monitoring it, and we let them feed and just to see the symptoms of the disease. And these are the results. So in the fourth week, we have 61% of that field showing symptoms of disease, and 39% of the field was healthy. Why I'm saying that's healthy? Just because they're not showing symptoms of the disease in that moment doesn't mean that the pathogens are not inside, right? Means that maybe they got infected later in the season, in the vegetative, in the reproductive stages, and then the pathogen didn't have time to express, to multiply in a load that can express the symptoms in the plant. So I think that actually 100% of this field was infected with the pathogen inside, but just like 61% at this point were showing clear symptoms of corn stunt. Just here some pictures that I took by drone just to show you guys this reddening of the leaf streaks again, especially in the upper part of the plant. Here again, you can see this reddening. It's just to mark the symptoms in case you guys see this in the field in the next years. How do you manage corn stunt, right? I think that's the question that you guys want us to answer, because I already show a lot of symptoms. I already showed that this can be a problem, but how can we manage this disease? So I have bad news for you guys, especially for me as a plant pathologist. There is no curative strategies that are currently available to manage corn stunt disease by targeting the pathogens. So this is not a problem that a plant pathologist can solve, because once we see the symptoms in the field, there's nothing that I can do. I can go there, I can identify, I can tell the grower, okay, you have a problem with this corn stunt. But I cannot in that moment say there's no product that you can apply to solve this. So that's the reason we need to partner with a good entomologist to try to solve this problem, because by myself, I can just identify the symptoms, provide diagnose information, but I cannot really provide any management strategy. I don't want to mention about the sources, the management strategy that we use to control. I'm going to let this for Dr. Ferris. But I just want to mention that volunteer corn can serve as a source of inoculum. What this means? Means that the pathogens that cause corn stunt, they can survive in volunteer corn if those plants survive from the winter. What is not very common for us here, but I just would like to mention. So this picture was taken in Carroll County in November 14, 2024, and this is volunteer corn. We collect those leaves here, this reddening of the leaf tips, and guess what? They have corn stunt pathogens. They have Spiroplasma conchyli, that's the main pathogen associated with this disease. And guess what? In November 14, the hoppers were thriving. We are using jacket in the field. The hoppers are not, and they're still fine. So if they're going to survive the winter, I'm going to let Ashley talk more about that. So the take-home message from my presentation is that there's no curative strategies for corn stunt targeting the symptoms, targeting the pathogens. So we need to prevent this by managing the corn leaf hopper. We have a wide diversity of symptoms, so we cannot rely just in what our eyes are seeing in the field. We need to send samples to a lab to identify at least one of the pathogens that are causing corn stunt. The plant is most susceptible on the vegetative growth stage. In Brazil, they used to say that if you manage the corn from from emergence until V8, if you cover that period, you control the leafhopper, you'll be good enough to not have impact on yield because the pathogen is not going to have time to multiply, even if you infect the plant later, it's not going to have time to multiply inside the plant. We need to eliminate volunteer corn plants because this can serve as a source of inoculum. The pathogens can survive in volunteer corn plants and serve as a source of inoculum for the other season. And we need to test and monitor corn stunt pathogens. It will be key for the management because we need to know what pathogens are involved here in the United States because this is going to be key for companies start to breed hybrids that are resistant to this disease. And we need your help. So I'd like you guys to please pick it up yourself and scan this QR code. We need to receive samples from around the state of corn stunt symptoms. So if in the next growing seasons you guys are seeing symptoms of corn stunt, please send to the Plant Disease and Insect Diagnostic Laboratory. Most of you guys are familiar with Jen Olson. She managed the lab and she has the capacity to identify those symptoms. So this is just an example of a sample that we received from New York State. And also contact me if you see those symptoms. I will be pleased to go to the fields, scout the fields, bring samples back. So you can contact both me or the Plant Insect Diagnostic Laboratory. And I'll take questions in the end. I'm going to let Dr. Ferris take over here. And that's it that I have for now. Thank you. Do you want these or the?
I'm going to use the hand microphone.
Okay.
One slide we had up there, how can you distinguish between more pollination and the kernels not filling?
I cannot by seeing the symptoms in the field. I need to bring back samples to the lab and see if the pathogens are inside the plants or not. Just by seeing the symptoms, I cannot say that's a drought stress that affects pollination, lack of water, or if it's corn stunt.
All right, before I lose too many of y'all, I'm going to go ahead and get started. So I am Dr. Ashley Ferris. You can just call me Ashley. I am the new Tom Royer. So if you ever worked with Tom, that's who I replaced. I've been told sometimes I have a little better personality. No, I don't know, but I really like Tom. Tom is very helpful. I really enjoy working with him. He is also retired, but I still can call on him to ask questions, which is really good. So I am the IPM coordinator and the cropping systems entomologist. So entomology wise, you understand what that means. I'm in the field looking at bugs, try to solve those problems. IPM coordinator, I want to mention that because of the mixed audience we have in here. So some of y'all that might be familiar with David Kearns, he's the IPM coordinator for Texas. I'm the kind of David Kearns, minus the dean role, thankfully, of Oklahoma. But it's really important that I have this role because I work with different commodity groups, industry partners, as well as with funding agencies to try and get funding for IPM programming that is specifically related to extension outputs and how we can help our producers. So having relationships with y'all is also very important for that because you're giving me the data that I can put forward to these large funding bodies to be able to justify why Oklahoma demands those highly competitive dollars in order to continue to fund those IPM efforts. So I started my position back at the end of January. I can tell you about my moving trip a little bit later. But just because Brian told me last night that I hadn't been doing this, I am a Texan. So I do have to admit that. He said he was proud of me for not saying that recently. So I'm sorry, Brian. I am a Texan and I'm proud of it. But I'm glad to be in Oklahoma. So we did conduct a first report. And when I talk about this first report, and when a lot of us do, I want to mention this insect, based on what we saw from distribution of historical data, has probably been in Oklahoma before, but we just didn't know it. We didn't know what to look for, and we just weren't aware of some of the problems because, like Myra and also Dr. Waidende said, you know, we didn't know that some of these symptoms could look a lot like the stress factors that we see from drought and other kinds of environmental conditions and also insects too. So teasing those apart can be really hard. So I want to mention why we have data from certain locations and not others. I was in a hurry to get this out and published as fast as I could because this is information that's really valuable to our producers as well as also to other kinds of professionals too. And so I actually reached out to Maxwell Smith and I said, hey, do you have anything down here? And he said, everything is burnt up at this point when I contacted him in August. And so we wanted to look northward so we could start to tell people in Kansas, hey, you need to be aware of what's going on. So we had some spotty reports. I'm going to first talk about what we saw in Oklahoma for corn leafhopper and corn stunt disease. And these spotty reports are due to a number of different kinds of things. Okay, so all of the states where we had this, we were kind of caught in the middle of our seasons. So that caught us off guard of knowing what is happening. So we do have gaps of information that we're trying to fill in. And that's where conversations with y'all is really important. But we have some producers that are saying, was it impacted at all? Because they probably planted early. And so they didn't have to worry about the insect or pathogen load at just the right time. I've also talked to producers where they're estimating anywhere between 30 to 60%, and in some really devastating cases, 80% yield loss. And a lot of this is going to be found in our later planted corn. It was hit really hard. Like I said, early season had little to no impact. And I'm delivering this message today not to scare us from planting double crop corn, but instead I want us to be thinking about what are some things that we can do to help our producers get their foot started off right so that we can make sure they have the best chance possible. And I think that there are some ways that we can do this. So we found it in field corn and sweet corn, irrigated and non-irrigated field corn. Haven't really seen a difference in either one in terms of severity of symptoms or populations of the insects. And I'm underlining sweet corn. I know I'm talking to consultants that largely work in field crop commodities, but I don't know how many fields that I went and looked at in field corn. And I was talking to a producer and they're like, well, all the corn's out of the ground now. They're just worried about volunteer corn coming up. And I said, did you see what that person just around the corner from your field is actually growing? There's sweet corn that's still up and it's highly infested with corn leafhoppers. So you do need to think about what's going on around you so that you can be aware of, I know we can't control what our neighbors do, but bringing that to the forefront of our producers' mind to let them know where there's other sources might be, they need to be aware of that. And then again, really teasing out those environmental versus corn stunt disease symptoms, really hard to do. And that's where that confirmatory testing is so important. So we didn't know what to look for. I think that's one of the big problems that we had. And this kind of brings up the question of, is this something that's going to be cyclical, like what we saw with the history that Austri and also that Myra shared? Or is this going to be maybe something that we're seeing more reoccurring? It's definitely more expanded into the US, which I think is concerning for all of us. But the last time we really saw it was in 2016 out of the Rio Grande Valley in Texas. And when we talk about South Texas, that is pretty much as south as you can get in Texas. We do know that there is populations of corn leafhopper that are still out. And so that is also a concern too to think about. The other issue is for coming from Texas, where corn in some places is a little more intensively managed. It's not always the case here in Oklahoma. And so that's another reason why it might have been so easy to miss this. And so the other issue then, of course, was the environmental conditions. And so this is actually from a field that Beck Johnson had me come and check out. So thank you, Beck. But it was one field where I looked out there and it was the first one he had shared with me. And when I got to that corn, I ended up having to call Beck and say, this corn, it looks like hell, excuse my language, but I was like, I can't tell if this is corn stunt or if this is drought or if this is the grasshoppers that are eating up everything at that point that we were at in the season. And so then he was able to show me and say, hey, check out these fields. I've got a producer that really, I like to call it babies their corn, So he could tell me that all the right inputs were in there. And that really helped us find some of these symptoms. Also, very warm. We didn't have a lot of rain. And so luckily we have this kind of data from the Mesonet where we can start to kind of piece together what might have happened historically and what some of those other factors were. Austri touched on Hurricane Beryl, jet stream, Greenbridge. I agree. Timing doesn't seem exactly right. We've had hurricanes and tropical storms in the past whenever we've also had this insect, and it hasn't brought it up this far, at least not in the numbers that it did this time, and at least not also to the extent that it's found earlier. I'm more interested in, was this possibly a green bridge? So what I've learned from being here is that it was a really mild February. We did have some producers that were planting corn towards the end of February. Even in some cases, I think someone told me they planted middle of February, which I thought was kind of crazy, but the soil temp was right, so they did it. And then we also had issues in Texas. So in Texas, we were actually seeing that there was delay in planting when it came to area around Abilene, San Angelo. And so maybe we built this green bridge, which allowed for those populations that were building up in Texas to come further up, because then they had kind of one of those short migratory events that could then lead them here to Oklahoma, where we had built the green system for them earlier than what they would have needed otherwise to be able to make it here and beyond. So those are just some thoughts that I have. All right, so when we're looking at this outside of the growing season, we are finding the corn leafhopper in Oklahoma counties as of last week. It has been cold, okay? But also we haven't had the coldest temperatures yet. I mean, I'm saying it's cold, but also I've sat at my house on the back porch because it's 60 degrees and nice outside, recently too. So we have also, like Myra said, confirmed that in volunteer corn that still exists out there, we do have that spiroplasma that is present. And we are monitoring these potential overwintering sites. Okay, so in addition to looking at areas with volunteer corn, we're looking at things like Johnson grass. And so I know whenever I had a conversation initially with a colleague, they said, well, Johnson grass will die too. At the soil, like above the soil level, yes. But what do they grow from? Rhizomes. They're under the ground. That's why it always comes back, So there is potential where these leafhoppers could get into the soil where it is much warmer and it's absorbing the heat also, because it's warmer, darker material, and then possibly have a water source to be feeding there too. Another thing that we have a lot of in Oklahoma and in Texas is eastern gamma grass. And so actually this is from a thing that Laura Goodman put together with the different grasses. So if you need help identifying some of these different things, just Google OK State and then the type of the grass or material that you're looking for, and you'll probably be able to find images that will take you through how to identify these. I'm an entomologist. This doesn't have any legs, so I really can't tell you much more about it than look at this picture. To me, it looks a lot like-ish Johnson grass, but also think about the structures that you're seeing here and what Austri showed y'all earlier. It makes sense as to why this might be attractive for these to use as just a site where it can feed. And then also winter wheat. So we've got, our wheat that's being planted into like where the volunteer corn is also then coming up. And so that's another area we're looking at, and alfalfa. Really just think about this as you could insert any other crop system here too that's out there and is green. That's where we want to be looking to see, are those corn leafhoppers coming out of the volunteer corn that may have been knocked back by freeze, going to an area whenever it gets on a warm day to where they can actually fly, go and hide out in that habitat and be able to start to maybe take up some sort of like nutrient resource to where they can survive later on. All right, so they're only known to reproduce in corn in nature. And I say this because there's a lot of things you can do in a lab or a greenhouse and sometimes force an insect to do a thing. So there are studies out there where they have been able to be shown in a greenhouse to be able to reproduce up to, I think, like 5 generations on eastern gamma grass, okay? But that's in a greenhouse in a forced situation. There's no evidence of that actually happening in nature. So also we know that they can lay eggs at a certain temperature range. Lifespan is on average about this, but Austri has found, we talked a couple days ago, that she's come across literature which shows that they survive for several months as well, like away from their host. So that's also a little bit concerning. And then there is evidence that, maybe a 50% mortality will be reached whenever they are exposed to 23 degrees for 3 to 3.5 hours. Austri also shared that she has had to toss them into a -20 for an extended period of time to actually kill them. So there's still a lot of information to learn. What I'm showing here is basically some weather temperature history data. It seems like we're likely in a warm cycle right now. Like I said, February seemed to be pretty mild. So depending on what's going to happen for us in these next few months, I think it's going to be really critical to determine, are we going to have this insect present? So like I said, other green material, that's a nutritional resource. We do have evidence from the literature that actually that spiroplasma, if that insect is infected with it, it kind of acts like antifreeze, where they actually have the ability to overwinter even better because they're infected with that. And that also points to that mutualistic relationship that the pathogen has with the insect. And so that pathogen benefits that insect, So that's another reason to be concerned and kind of aware if we've got that pathogen still out there, we have infected ones, they may be able to live a little bit longer in a little bit colder temperatures than what we anticipate. So big questions. Will the corn leafhopper be in Oklahoma at corn planting time? We're going to find out. If not, how much less ground, though, do they have to cover to move from Texas into Oklahoma in 2025? And I say this because when I drive back home and see family, I see how much volunteer corn is along 35, all the way from Denton down into Central Texas. I'm working with colleagues that are in the coastal bend and in true South Texas too. There's a lot of volunteer corn down there. So we might be moving that gradient a little bit higher. They have less travel that they have to make to actually make it to us. So bad news, right? But don't panic, okay? We haven't had our coldest part of winter historically, at least so I've been told. So hopefully that's going to change. So that's one thing I want y'all to hold on to. So be praying or doing a dance or whatever you need to do to make sure that it gets cold enough. But we need to know if it can become established in Oklahoma. All of the studies that we've talked about, none of them have been done here. Few have been done in Texas. We have very different microclimate here in Oklahoma. We have different agronomic practices, agroecosystems, and other kinds of conditions and factors that we need to take into account. And so I'm really making a push for this because we get a lot of data that seems to come from states that neighbor us. But one reason Myra and I have been very proactive in trying to go ahead and put OSU out there on these things is because even though I'm a Texan, I know it kills me to tell an Oklahoma producer, we don't have that data, but we've got it in Texas. It'd be really nice to have that data from your own backyard, right? And then we can speak to what actually is expected to happen here. And so we can share that information with them as well. And I think it does matter because there are ecologically a lot of differences that do relate to insect behavior and insect ecology and how this pathos system is probably going to behave. So we really need your help with monitoring for the cornleaf hopper. And I'm going to talk a little bit more about what we're doing for monitoring in a moment and how you can help. So what are some ways we can prepare for the cornleaf hopper? Of course, the first thing everybody thinks of is like, oh, what about resistance or low or reduced susceptibility? We don't have any currently on the market here in the US. That is something that I'm aware of from different talks with different industry partners that is in the works. People are trying to tease out patterns of, is there possibly a variety or a hybrid that's available or a variety available right now that has some sort of reduced susceptibility. And so those are things that I know are conversations that are happening. And so talking about those patterns is going to be really important. Manage volunteer corn. So grazing, that's one way that it can happen. We also can have it happen by tilling, which I know can be, depending on your preferred practice, may or may not be something you're interested in. And then also freeze. And so here's one field that has been knocked back due to freeze. But like I said, when we found corn leaf hoppers recently, it was in corn that was frozen and the leaf hoppers were still alive and in it. It'd be interesting to see if they're still in there though, since we've had these warmer days, which is going to increase that insect activity to where now they're probably going to want to fly and find that green nutritional resource. Also, so Myra kind of pointed to this, if we manage this volunteer corn, we're removing the reproductive hosts, so we're hopefully not adding to the population, and we're also having that pathogen reservoir be knocked out as well. So best advice right now is plant early. And I know that kind of contradicts the idea of this double crop corn, but there's also other things we can do to protect that too. So I'm not saying do not plant double crop, okay? Just plant as early as you can and be aware as to what's going on. Also, you need to get that corn past the V8 stage. And I've actually had conversations with David Kearns about this when we were reading through different literature, thinking about what should we do. And there's also been times where we've both said, maybe it actually should be through to V10. But right now, the message we're really communicating is get it through that V8, and then you'll have a good chance of protecting it to get some sort of yield. So the other thing you can do is a high seed treatment rate for your insecticide, okay? I know that this is going to be a price point that's going to be something to consider in your management practices. But if you're planting late or double crop corn, you really need to use Cruiser Max Corn 1250, or you need to use Poncho 1250. And yesterday when Steve was talking, the person from Tennessee, my colleague, he was sharing, that Poncho seems to have better rain resistance, so like if you have a rain come in, it's not going to wash away completely or as quickly, has a little more staying power. So it seems like 2 to 3 weeks is what you can expect in terms of protection from these seed treatments, but it's a really good way to get things started for you. I am pointing out that if you're interested in wanting to know more, and then I would say some of y'all were asking me yesterday, were there different chemicals that they're using in Brazil and Argentina? Number one, some of the things that they're using there are things that we cannot use here because of the EPA. So take that as for better or worse, however you view it. So there are some limitations, but if you're interested in what those are, you can find them in that paper. I'm not proponing that you use those things, but just so you can see what other chemistries that are being used. They also, though, are showing in Brazil and Argentina that there is resistance to the active ingredients in both of these things. So understanding what population we possibly have here is going to be really important. So sending and sharing samples to some colleagues that are doing the haplotype work. I have a molecular background, but my focus really, like Myra, is being in the field and working with y'all directly. So I'm sharing that with somebody else that can get the information out for us quicker. All right, so scouting for corn leaf hopper is going to be very, very important. So basically, if you're not using one of the 2 seed treatments above, you need to be scouting at V1. And then if you are using one of those high insecticide seed treatments, you need to begin at V3. And you need to scout regularly and keep up with our pest e-alerts. So I'll have a link here in a little bit that shows you how to sign up for those. But that's how we're hoping to communicate mass wide to be able to share what's coming up and coming down the pipe. And also know what you're looking for, okay? So you really need to be aware of what you're finding in the field. Make sure it's actually a corn leaf hopper. As an entomologist, that's very important. I know not everybody studies bugs, but the good news is with this one, you've got those 2 little dots between the eyes that are something that can help you. You're probably gonna need like a little hand lens with you in the field. And you may wanna take some vials or something with you that you can catch one in because they do move pretty quickly. All right, so not every leaf hopper you find in the field is going to be one. The other thing I want to mention, I think, Mario, you're pointing at these. Okay, so I have, if y'all want to see just how small they are, Braden, I'll let you hand it back. I'll take one to their side of the room. Okay, if you want to see how small these guys are, I've got some, so I'm an entomologist, so I've got a colony of these in my lab right now. They're contained, so don't worry. But I'm keeping them for research purposes, but I pulled some out so you can see that they are pretty tiny. So having that hand lens is going to be really important. But being sure you know what you're dealing with, so you're actually spraying what you're supposed to be spraying, is an important part of IPM. All right, so when we're talking about the field identification, here I just want to show this picture of the nymph that we've got. They are wingless. They're more yellowish in color. They can be brighter. So as Austri said, as they get older, they're going to darken over time to some sclerotization. Again, very, very small whenever we're looking at the size of them too. And when you're looking in the field, depending on the size or the growth stage of the plant that you're at, you might want to start looking underneath the leaves as well. They like shade, just like we like shade. And they have a very different behavior than the last invasive pest that we had. So during my postdoc, I worked with the sorghum aphid in Corpus Christi, which is part of the coastal bend of Texas. And so like Steve was saying about the pains of the honeydew and everything, I know exactly what he's talking about. But this guy is super easily disturbed. Aphids, lazy, they're going to stick to the underside of the plant until they grow, some wings, and they actually do take off one of their migratory events. But this one is much more challenging to scout in the field. So have your eyes trained. So these are some pictures of me and Myra, and actually Seth, you were with us for these too, when we were some producers, where we have them deep in the whorls here on these slides. If you were to look at it, those 2 little dots, let's see, oh, nope. 2 little dots though, that are like here on this slide. I don't know why I can't get the pointer to work. There we go. Those are the corn leaf hoppers. So really getting down there and looking at them is very important. And then here's just a close-up image of those 2 dots right here, the corn leaf hoppers looking back at you. All right, so you're going to want to start at emergence, continue through V8 for your scouting. Check those edges of field borders. I think Myra convinced you of that. Where to look depends on the growth stage though. So in the whorl is important, but also on the underside of plants going to be really important too. And to sample deep in these whorls, use a handheld vacuum cleaner. It has a clear canister on it. I'll show you what one looks like that I recommend here in a couple of slides. So here is just me at a plant that we've got at Sweet Corn. And so you can see some of the corn leaf hoppers kind of moving around, just how quickly that they move, and also just how small they are. So also check those collars too, whenever you're looking for the corn leaf hopper. And then when I was talking about that vacuum cleaner, seriously, something off the Amazon, 20 to 30 bucks, I think is like what you'll spend. Some people are modifying them with pantyhose. Unless you're keeping them to sample them for different research purposes, you can suck them all up into one, so it doesn't really matter. But again, just how small they are, having that clear canister allows you to see if you're sucking any up. You can just hold them over the whorl of the plant, and then you can sample them pretty quickly there. Another way is sweep netting. This is a pain in the butt in corn, okay? So I'm just going to admit, much easier to do whenever you are talking about this, about using the vacuum cleaner. But the sweep net is an option. You can cover more ground more quickly, but also think about the behavior of that insect. Are you likely to catch it as efficiently? Because it's darting around, and the way that it darts is very different than I think what our brains want to process of the direction it's going to go. And so if you are sweeping, you can do this probably up until about the V5 stage, walk over the rows, make sure that you kind of smack the plant with the top of your net, and then always fold the end over really fast. Sweep netting is always easier looking than what it really is, okay? So until you do it yourself, you don't really know. And then also V5 to V8, you can do it. You're going to have to sweep up and down the stalks of the plant. Again, it's quite a workout, but it can be really challenging. So again, vacuum cleaner, I think, is going to be really easy. And then passive monitoring. We do have to advertise this because we do have to think about time constraints that people do have. And this is actually the monitoring system that we're currently using and we have used in the past too. So placing out one per field site, checking them weekly, maybe even more regularly, depending on how much you want to check them. Adjusting the height of the trap to the height of the corn. Okay, so we are using some shepherd's hooks here of different heights. You can also, though, use a wooden stake and staple it to it. Yellow, based on literature, is attractive to corn leafhopper. I've had a couple of people approach me about, like, what if we built this new trap or try out these different kinds? I'm not going to waste my time with that right now, okay, in terms of color. Color works. We know that they're attracted to this yellow color. And you can see that we've got a number of different corn leafhoppers here on this. And this is actually one that's not, we've got some that are full of corn leafhoppers. So this is a nicer one to look at. All right, so exoskeletons, be sure to be aware of those. Those are going to be those cast skins, basically, of our insects. In here, I think we've got an adult right here, if you want to see the size of it, a nymph down here, so a very early nymph, so the size really can vary. And then also that honeydew city mold. The other thing to note about this is, yes, this is a piercing, sucking insect outside of the pathogens. It would take a very high number for them based on their feeding alone to cause damage. But the problem with this photosynthesis, problem with this honeydew and sooty mold is actually photosynthesis issues. And so it can't actually get the sunlight that it needs to be able to generate the energy it needs for the plant too. So in thinking about this, I walked into a couple of fields. It was actually worse than walking in with the sorghum aphids. I walked out, my hair was coated in honeydew. It was absolutely disgusting. And it's just because corn's so much taller than sorghum. So thank you. All right, so protecting corn through the V8 stage, why is that? Myra kind of talked about timing and how long it takes these pathogens to circulate inside the insect, be able to build up and then be able to be transmitted, and then the delay in that transmission actually resulting in the symptomology of being present or showing. You also need to think about what happens at about the V7, V8 stage is the number of kernel rows is being determined. And so that's also going to be another important reason as to why we want to protect those stages. There's no economic threshold. You should assume, because of the efficiency of this vector, of this insect, that if you see one, that it is infected and has the ability to be infective. So that is why we like to promote the idea of spraying whenever you see one. All right, so what are some chemical control suggestions that you can use? So some things that are being suggested right now are pyrethroid, as well as chlorpyrifos and dimethoate. I will say that out of the coastal bend, whenever they decided to do a later season spray, and what for them, at least it was later season. So what they found was that they could get protection for about 7 days, good protection with a pyrethroid. And I'm saying this because you're probably going to have multiple spray events. Okay. So that's another conversation to have with your growers about the cost of the investment on some of these things. Chlorpyrifos and dimethoate, they worked fairly well also. But for all three of these, especially pyrethroids, you need to be cautious because you could flare those spider mites. Similar to what Seb was talking about yesterday and some other cropping systems, spider mites are one of those things that, an unexpected problem that can be really unpleasant really quickly. Some other suggestions that are coming out based on evidence from studies out of Brazil and Argentina are also the use of Savanto Prime, which Seb also endorsed yesterday in talking about what was used. So these are two that actually we use for the sorghum aphid or sugarcane aphid, however you want to refer to it. The one thing is there is a price point with that. We don't have evidence just yet of it working here in the US. The idea of in furrow, like what Seb talked about yesterday too, would be very important for getting that lasting effect of the Savanto Prime, so that lasting protection. So that's another thing to be thinking about. Something that I would really like us to test here at Oklahoma. And so I've got some field trials that I'm trying to work on to test out some of these things in the future. So again, multiple treatments are likely across the season, depending on when you get that infestation of those corn leaf hoppers in your field, if we get them at all. Okay, so this information on this slide It is very much, please take it with a giant grain of salt, okay? I actually debated on whether or not I should share this with y'all because I don't have statistical power right now to really elucidate things. But I think it's important to show what we've done to kind of test out some different compounds and chemicals to be aware of. Primarily, I want you to know that there are a number of things that are labeled for corn leafhoppers and corn, except for that very first one, okay? Plinazolin, that is a product that Syngenta has talked about for a good while. It's not something that's released just yet, but it is something that I think most people are now aware of, which is why I'm also okay with naming it in this talk. I did try these other types of chemicals at different rates too. Kelly says was very helpful with me on performing this study. But we were doing this in a lab setting, okay? So that's the other caveat to this. We basically built these little Petri dish arenas that we put a leaf dip that we dipped into the same adjuvant, reduced the amount of each of these chemicals to be representative of what would be found on a foliar application. And then we just needed to have more samples and able to be able to elucidate some statistical significance. So I'm going to show you some patterns that we found, okay? And so it's just to demonstrate that there are products that are out there and we saw some sort of toxicity with them in a lab setting. So for plinazolin and Stewart at a 6-ounce rate, we saw mortality of the corn leafhoppers in our little arenas at 24 hours, okay? For dimethoate and Savanto, we found mortality within 48 hours for the majority of the corn leafhoppers. When I came back and did our 72-hour check, all of those were done, okay? They were all dead. And then Elevest and Hero, we saw mortality within 72 hours. These are the ones that I can talk about a clear pattern that I saw. Again, no statistical support for this. And again, very, very much preliminary data in a lab study. I want to share this with you though, just because y'all need to start looking at what are different labels, different options, different price points also too for the intensity of management that you want to do. So that's just something to be aware of. What I really want to do is take that information though and put it into a field trial so we can translate that, see how it actually works in the field. But of course, it's all going to depend on, do we have the insect here in Oklahoma? I could do more lab trials because I've got a colony. I actually have to kill them off sometimes because they reproduce really, really well. But we do have a colony of corn leafhoppers. And so there are opportunities for other kinds of studies to happen as well. All right, so within the context of Oklahoma and agroecosystem and practices, I really want to be sure that we're generating this data that's relevant to our producers here. All righty, so what we're also doing is we're doing trainings for diagnosticians. We're doing this through the Great Plains Diagnostic Network, where we're going to be doing a webinar in January and some hands-on workshops for how to do some of those pathogen detections. We also, Mayra and I, teamed up with colleagues at Kansas and also at Missouri to propose a working group with the Southern IPM Center. We gathered the support of 18 different states in the U.S. That was a team of entomologists and also plant pathologists in order to help create this group where we can reduce duplications of efforts, finds ways that we can maximize resources and share knowledge for how we can manage this. So kind of a big information exchange. We'll find out if this is actually funded in February. And at that time, we're ready to talk to and see how does the group want to interact with industry professionals, bring in producers, crop consultants, different kinds of factors in there too, so that we can be a team that's all working together on this. And we've also been working with colleagues at Brazil and Argentina where this is endemic. And so we're learning a lot of information from these individuals that have been really helpful in working with us. So what we're doing is we're having this active monitoring network. We received a very generous donation from Trace for a number of sticky card traps. And so we have plenty available to where we can extend this network. And this is where we really need y'all's help to do this. So we've had some really good extension area specialists as well as educators that have been very, very helpful in us getting these traps out, but we need to extend this network even more. So I have up here this little QR code if you are interested in helping, which I really hope so, because I know you're going to come and talk to me later on. So raise up your phone and get this information so you can contact me too. Let me know if you have someone, a cooperator that we can place a trap out at, or if you'd be willing when you're going out to these sites too, to go ahead and take a picture of this trap and share it with us too. We're just asking you to place a sticky card, report back the information that you see to us, and then tell us as soon as possible. That way we can generate out those alerts and get all of us on the same page. So I also have that contact information thing here again too. You can sign up though for pest e-alerts. You can go to the webpage here to do that. If you also just Google OK State Pest E-Alert, it will take you to the page that I've got over here on the right-hand side, and you can sign up for the Pest E-Alert subscription. So I plan to have the first Pest E-Alert by mid-February to let you know what we've seen in the field, where the update is on that. Also, give you some updates on any kind of management guidance, anything that might have changed or anything else we may have learned. And then also information on how to access EdMaps. This is where I also want to toot our horn on what Oklahoma has done. We were the first ones to reach out to the Southern IPM Center and say we want to communicate with our producers using EdMaps, which was helpful to both Kansas and then Texas that followed suit after us of how they also want to communicate the information. This is exactly what was used for the sorghum aphid. So we know it's a system that some producers and likely y'all are also familiar with too. And I just want to give a big thank you to the people that are already helping us with this monitoring effort. So we have some upcoming grower meetings. These are just two that I wanted to list and share. So if you're interested in them, then the contacts are on there too. But for the one at Carnegie Cotton Gin, we're going to be having that kickoff in the morning. It will have a lunch with it too, and then we'll disperse after lunch. So it'll be a morning activity. And then at OPREC on February 13th, we're going to be doing a crops clinic, and Submit will be the one that you can ask about what else is going to be talked about there aside from the corn leaf hopper and also the corn stunt disease. I also want to mention that we've teamed up with the suction trap network. So that's how we've gotten a lot of the data about what's going on just north of us. We are anticipated to have a suction trap in Oklahoma. So we'll be extending that southern range, and having that here also offers us a ways to get alerted about what other kinds of wind-borne insects are possibly coming in the future to be invasive. So hopefully we can get a better idea of what's going on then. So with that, I've got two other QR codes. This one, again, my contact info. This one up here, if you want any resources, you can click on that. Then the resources that I have cited and referenced, as well as links to other types of corn leaf hopper references, are going to be here. It's going to take you to a Google Drive that you should be able to access. Okay, so you can get those references for yourself. So with that, thanks for your time. I don't know if we have time for questions or anything. All right, thanks.